So today we will talk about different plant breeding procedures that are being used in the breeding of crop plants for as far as self-pollinating and cross-pollinating crops are concerned. So to understand this concept, first we need to look about what is a segregating population. So to understand the concept of segregating population, first we need to understand what is hybridization. So, what is hybridization? Basically, hybridization is a phenomena in which genetically different parents are crossed, which results in the formation of a segregating population. And this segregating population contains a genetic diversity which can be used to improve crop plants through breeding. So, as you can look on your screen on the left side, there is a figure which consists of a parental generation. If you look closely, that one parent is having a tall plant height while the other parent having a shorter plant height. Both contains different sets of genes. Both are genetically different parents. That's why whenever these two types of plants are crossed, they will always results in the form of a segregating population. So whenever the parental generation of these types of plants are crossed, they results in the formation of F1 generation. So we will never do selection in F1 population because if we look at the morphology of the F1 population, all plants will be look similar, although they are heterozygous genetically, but visually we cannot distinguish in F1 generation. So that's why we will never perform selection in the F1 generation. So after F1 generation, when we get an F2 population, so in this population, as you can see, there is a segregation concept. You have all different types of height of plants. For example, some plants can be a very tall plant height, just like the parental generation. Some plants can have a mediocre plant height, while the other plants can have a very short plant height. So here in F2 generation, you will get a segregating population, and you can start selection from F2 generation. So now let's have a look on the definition of segregating population. A segregating population is the genetically heterogeneous population that results from hybridization between parents that are genetically different from each other. So in plant breeding, how to handle these types of population? So normally there are three uh, different breeding techniques that are being used to handle segregating population. The first technique is known as pedigree method. The second one is bulk method, and the third one is the back crossing method. So all these types of technique are being used to create new and pure variety. In this lecture, we will discuss about bulk method. To understand the detailed concept of bulk method, let's divide the bulk method into different terminologies. The first one is the bulk, which means harvesting of all the seeds from all plants from one generation, use those seeds to grow the subsequent generation. The second one is the delayed selection. Basically, the bulk method is divided into two major categories on the basis of delayed selection. The first one is the short term bulk method, in which we delay our selection until F6th, F7th, or F8th generation. But whenever we deal with the long term bulk method, we delay our selection until F30th generation. The third point is the natural selection. It means breeders do not impose his selection because it is the nature that allow the plant to be selected naturally. So the fourth point is the artificial condition. It means the plant breeders can only create some sort of environment in which a plant can be grown naturally or a plant can be survived naturally. So when we combine all these terminologies together, let's have a look on the a proper definition of a bulk method. So basically, it is a breeding technique where seeds from F1 and the subsequent generations are harvested in bulk to raise the next generation or subsequent generation until homozygosity is achieved in a population. and this homozygosity should be achieved in a naturally way, or we can say that we should perform this type of selection until the desired objectives have been achieved, right? So let's have a look how bulk method of plant breeding actually works. In the first year, select genetically different parents and perform a hybridization process, which results in the formation of F1 generation. And remember, we do not perform selection in F1 generation. So what we should do, we should harvest all the seeds collectively, and in the second year we should plant all the harvested seeds in a spaced planted manner. By space planting means we should let the two plants be separated from each other so that every plant will have a equal chance to grab some water, some nutrients from the soil, so that every plant can produce a healthy seed. So again, at the end of this year, when the crop is matured, we should harvest all the seeds from all plants in a collective manner, or you can say the bulk them together. And similarly, in third year, what we do, we should plant all the seeds which we have harvested from the second year, use them to grow the F2 population. Again, you can see from the figure, it is very clear indication that here the segregation comes. So whenever we're dealing with the bulk method, we should not perform selection in F2 generation. So again, in F2 generation, we will harvest all the seeds from F2 generation and let them grow in the fourth year so that we can have our F3 generation. Remember one thing, that again you need to plant all the seeds as a space planted manner. You should allow the plants to grow naturally, to harvest maximum nutrients from the soil, to harvest maximum water from the soil, so that they can show their true potential in a true manner. So again, in the fourth year and from the F3 population, you should again harvest all the seeds from all plants and then move to the fifth year. Again, you should plant all the seeds which you have harvested from the previous generation, so in this generation, and let the plants to grow naturally. Again, harvest all the seeds from the plants, and in the sixth year, again you should plant the F fifth generation. Again, you should not perform selection, but rather you should harvest all the seeds together and let them grow in the next generation, which is the seventh year, and technically it is the F6 generation, right? As you can see, we are not performing any kind of selection. We are just bulking the seeds from every generation and letting them grow in the subsequent generation. So here you can see that we can divide our bulk method into two different forms, as I discussed earlier. The first one is the short term bulking, which means we have to stop the selection until F6th or F8th generation. So later on we will talk about that how long term bulking method works, but at this moment let's focus on the how short term bulking method works. Until now, we have discussed about bulking of the seeds and dealing of selection. So from now onwards, we are incorporating natural selection and artificial condition. So for an example, uh, we are performing a short-term bulking method to develop a variety that is resistant from the disease. So in this example, we are taking an example of a wheat. For example, we are trying to develop a new variety of a wheat that has some resistance against a leaf rust. So as you recall, that in the first year we have selected variety A and variety B, let them cross together by performing hybridization to have the F1 seeds, and from F1 seeds in the next year we have planted the F1 generation. So whenever we planted the F1 generation, we should also plant any wheat cultivar that has susceptibility for the leaf rust. So it, according to this, we are performing a natural selection. Remember that. So as you must have known that the disease always prevails whenever a disease triangle complete. So for the disease triangle, what should we need? For the disease triangle, there should be a pathogen, there should be a host, and there should be an environment. Okay, so suppose the environment looks good for the disease, so in this way the disease can be spread easily. And sometimes when there is a not too much good environment for the disease to prevail, so what we should do, we should apply some pores of the leaf rust on our F1 generation. In this way we are letting the plants to survive naturally. So what will happen that the spores of the disease spreads all over the field, and eventually only those plants which have a natural ability to survive under this sort of condition will produce seeds. So remember that you should always harvest seeds from those plants that have survived. Or you can say that the other plants which are under heavy attack of the disease, they will not be able to produce seeds. So in this way you have selected the seeds plants naturally. So use the seeds which have survived the attack of a disease, and let those seeds to be used in the next generation, which is the third year, to follow the F2 population. So in the F2 population, we should again perform the same procedure which we have applied in the second year, that we should again harvest only those seeds which have the ability to survive naturally. Here you can see that again in the F2 generation, we will apply some sort of disease spores, which is, remember, which is an artificial condition. We are giving artificially condition of the plants to survive naturally against a leaf rust disease attack. This sort of procedure we will be proceeding until F6 or F7th generation. Again we need to perform the same procedure which we have performed in the previous generation. Create an artificial condition, let the plant to survive naturally, and harvest seeds only from those plants that have the ability to survive naturally and to produce healthy seeds. So you can see that we have reached the seventh year and the F6 generation of the plants to develop a new variety that is have ability to perform better against the leaf rust. So in this generation again we will harvest all the seeds from F6 generation and let the grow in the next year to develop into F7 generation. Okay, you can see, have a look. So this is the year where you perform a progeny test. It means you should identify a good looking lines, and from those good looking lines you should select the plants that are according to your will, or you can say that have the natural ability to perform better against the leaf rust. So in this generation you are performing selection, right? So you should harvest seeds only from those plants that have ability to perform better that against leaf rust, and beside this that have also ability to perform good or better yield. So you should harvest the seeds from the plant separately, as you can see, from this plant and from this plant and from this plants. Use the seeds from only these plants to move forward into the F8th generation or the ninth year. So as you can see that we have harvested seeds from this plant, and again in the next year we have planted the whole line from the seeds of a single plant. Again, we have selected the seeds from this plant and we planted another line of the seeds of that plant. Similarly for the third plant as well. Or you can see that we should grow individual rows from the seeds of each plant. Again, uh, you should have some competition between the selected lines. So as you can see from this figure, we have selected two better performing lines from the three, right? So we should selected the seeds now from only these two plants. So we have selected two lines out of three that are performing better. But in this example, I am taking line number one as a complete example to proceed further. So again, we have selected line number one seed from this example. What we should do, we should now proceed for the 10th year to perform a preliminary yield trial. So in this example, we have taken the seeds from the line 1, and then we should make a competition with the already available check cultivar. But remember one thing, in the preliminary yield trial, for example, if you are performing breeding in the southern Punjab region of the Pakistan, so what we should do, we should uh, make a competition of a newly developed line with the already available cultivar of the southern Punjab region of Pakistan. For example, in this region, in the southern Punjab of Pakistan, we have a wheat cultivar which is named as Nawab 2022. So, in the preliminary yield trial, we should make a competition with our new line versus already available cultivar. So, if our new line perform better as compared to the already available cultivar, in this way we can say that we have developed a new variety by following a short-term bulking method. But beside this, because sometimes we are going to develop variety not for a specific region, we should perform a multi-location yield trials. So normally a multi-location yield trial uh, lasts for 2 to 3 years. So as you can see that here it is an example of region number one. That let's say this is an example of a southern Punjab region of Pakistan. This is the second region, which is the, you can say that, upper Punjab of Pakistan. For example, in the upper Punjab of Pakistan, we have some renowned cultivars. For example, Chakwal 50 is another wheat variety, or we can say Chakwal 87 is another wheat variety. Wafaq is another wheat variety of the upper Punjab region of Pakistan. So in the region 2, we should make a competition with our newly bred lines with the uh, specific variety from the specific regions. And in the region 3, let's say it is KPK province of Pakistan, and from the KPK we have certain wheat cultivar just like Peer Sabak 2013, Peer Sabak 2015. So we should have a competition with our newly bred wheat lines with the Peer Sabak 2013 and Peer Sabak 2015, and so on. If our newly bred line perform better in all the regions, so in this way we can recommend that a variety has a natural ability to perform better in different regions, so we can release that variety in all those regions. Or in otherwise manner, for example, if our variety performs only better in region number 1 or region number 3, for example, so in this way we can say that our variety should be released as a actual variety or as a cultivar in region number 1 and region number 3, right? So at the end of 13th year, if you are able to perform good results, we should perform a seed multiplication or perform a seed certification task in the 14th year, so that we can have enough seeds for the farmer, so that they can develop, so that they can use those seed to be planted in their lands and harvest maximum out of it, right? So in this way we have developed a wheat variety by following short-term bulking method. So just recall, I have I have mentioned earlier that there are two different kinds of bulking method as far as selection is concerned. The first one is the short-term bulking method, which we have discussed earlier, in which we are performing selection, or we have delayed selection until F6 or F7 or F8 generation. But in the long-term bulking method, we should delay our selection until F30th generation. So normally, long-term bulking method is not followed around the globe. Normally, a short-term bulking method is being followed, so that you can have a variety on conventional method in 13 to 14 years. So let's have a very look about the advantages and disadvantages of bulk method. So normally, as far as advantages are concerned, it is it is a natural selection, because whenever nature selects nature selects the best thing that have the ability to perform better in a long term of time period. So this is the main advantage. And the other, you can say, that disadvantage is that the breeder has limited interference in the selection of plants in the earlier generation. Although breeder can have his say in the later parts of the generation, but in the early generation from F2 to F6, F7 generation, breeder do not impose his selection, because everything will be selected by the nature. So this is some sort of you can say disadvantage, because sometimes it can happen that a potential genes can also be lost in this kind of breeding method. But overall, it is a relatively good method. Normally, in Pakistan, we follow a bulk method or a pedigree method to perform a development of a wheat variety or other plants as well. So, I hope you have learned something from this lecture. And in the next lecture, we will be discussing about the other two procedures, which are pedigree procedure or a back crossing method. Thank you.